Okay, come on in, anyone and everyone who's ready to go for day 2 of the Phosphorus Forum. Welcome, everyone. I hope everyone had a hearty meal with no leftovers last night. It's great to be here and see everyone again. And we're going to have a really interesting session about, so often the neglected part of the food phosphorus cycle, which is dealing with food waste. It's, as we heard yesterday, if you were in that session, 30–40% of food is wasted. We need to do something about that. So I'm pleased to introduce the moderator and chair of today's session. Dr. Treavor Boyer is professor in the School of Sustainable Engineering and the Built Environment at Arizona State University and program chair for environmental engineering. And his research spans water quality, wastewater treatment, and numerous projects with innovative applications of ion exchange technology, such as PFAS removal from groundwater and nutrient recovery from source-separated urine. And so he's going to take over and get us rolling. Treavor, take it over.
All right, thank you, Jim. Great to see everybody. I just have a couple slides now, and then I'll have a few more slides when session wraps up today to put things into context. So here, this is our phosphorus flow diagram that many STEPS researchers would be familiar with, and probably many here in the audience of just thinking about how phosphorus moves through the environment. I've highlighted the sort of food component, thinking about both the inputs and the outputs to it. And, from a STEPS perspective, which is kind of the academic, component of the Sustainable Phosphorus Alliance, what are we thinking about in the context of phosphorus sustainability in food? Well, we want to increase phosphorus use efficiency, at the agricultural level. We want to decrease phosphorus pollution and runoff. A lot of talks today are going to focus on this third point here of really maximizing P recovery from waste streams. And, we've had kind of like parallel work as well in terms of thinking about the contaminant component. And then a piece where, we might touch on a little bit in the panel today, and then I have a few slides when we wrap up today, which is, really thinking about phosphorus use efficiency, through our diet, through alternative proteins. And so that's some background. One other slide I wanted to include here, this work came out of a phosphorus research coordination network that Jim led. That was kind of some of our earliest work on phosphorus. And this was a paper where we were thinking about, how do we transition from the sort of linear phosphorus flow to a more circular phosphorus flow. There's a lot of different models out there in terms of the sustainability transitions literature. And this one we felt helped us, articulate a vision for phosphorus sustainability where we have these 3 levels that are interacting, right? You have your niche innovations happening, and some of the talks today will talk about, not necessarily just startup companies, but early stage technology, where is its role? Then we move into the actual sort of regime, which is, kind of the status quo of what's happening now and looking at innovations there. I mean, I think the talk from the North Carolina perspective on sort of the status of food waste is a good illustration. And then we have these broader landscape pressures. A good example for us today would be mandates to, divert food waste from landfills, other sort of CO2 footprint considerations. And so I see all 3 of these levels interacting, and they really set the stage for the work that we're thinking about in here. And so now I'll introduce our first speaker. Our first speaker is Matt De La Hoysa. I'm sorry if I did, how's the last name? Good enough? Okay. So Matt is commercial director for BTS Bioenergy. He brings over 20 years of experience in developing environmental and circular economy programs. His educational background includes a Master of Science in Environmental Management and Policy from the International Institute of Environmental Economics at Lund University, Sweden, and a Bachelor of Arts in Economics and Environmental Studies from Emory University. So thanks, Matt, for joining us today. That was a great discussion. I'm happy now to introduce Christine Whitmere. She is in the Organics Recycling Lead and Recycling and Materials Management section of DEACS. She holds a degree in environmental science from Auburn University. Before joining, Christine served as an environmental programs supervisor for Henderson County, where she taught backyard composting workshops, piloted composting programs in 6 elementary schools, and helped construct small type 3 compost facilities at a transfer station. With a strong background in environmental education, sustainability, and materials management, she is dedicated to advancing organics recycling initiatives and food recovery in North Carolina. Christine has really a great set of slides. I've looked at those, so I'm excited for all the information you're going to share.
All right. Thank you so much for having me today. Again, I'm Christine Whitmire. I'm the Organic Recycling Team Lead for DEQ, and I'm out of the Division of Environmental Assistance and Customer Service. And I always like to start off that I am non-regulatory, and we provide a lot of great assistance across the state. We have permitting assistance in each section, in each regional office. We utilize retired engineers and scientists to help companies reduce their waste through our Waste Reduction Partners program. And we also manage the regional offices across the state. And finally, I'm in the Recycling and Materials Management section, the State Recycling Office. And that's also known as RAMS. We have a ton of acronyms, as y'all do too. And again, we provide technical assistance to local governments, recycling businesses. We offer grant programs. We collect statewide data to release reports, and we host events and workshops. And I know y'all heard from Jeff Constantino yesterday, so I won't get too much into the whys, but really talk about kind of the organics recycling infrastructure and collection programs across the state. But as you know, food waste is a huge problem across the country, and in North Carolina, ReFed estimates over 2 million tons of wasted food was produced in 2023, and about 40% of that was landfilled. And while all that surplus food is equivalent to $12.4 billion, and while all that waste is happening, 1.6 million people face food insecurity, including over 438,000 children. So we're really working to not only recycle the food waste, but prevent it from happening in the first place. And while, again, all this waste is happening, our per capita disposal rate is outpacing our population growth. And the two largest regional landfills in North Carolina, which accept about 25% of the state's waste, may reach capacity within the next decade. And the state recent solid waste materials management report estimates that the state has about 28 years of waste disposal if we are to continue on that rate. So again, we are trying to save that landfill capacity as much as we can. And fortunately, our local governments have reported an increase in their recovery rates. And this past 2024, local governments have recovered 1.65 million tons of materials for recycling and composting. And the recycling is shown in blue, and that also includes food waste. And then the green is yard waste. So my team works to understand the current infrastructure and identify gaps in the state that we can then address through our grant program. And I'll explain a little bit about how DEQ, the state, permits these facilities. North Carolina has about 385 organics recycling facilities. And this number does include facilities that have a permit, but are still in construction, or have a permit and are inactive. They did not report any waste that past fiscal year. But this number does not include exempted type 3 facilities. In 2019, our state released new state compost rules where facilities under a certain threshold do not have to get a permit if they follow state rules and compost a certain amount of feedstocks. And this number also does not include anaerobic digestion at wastewater treatment plants or our animal mortality composting permits that the Department of Agriculture releases. And so at DEQ, we kind of have like a handshake where if your feedstocks, the majority nitrogenous feedstocks determine who permits you. So if you're 50% or more of food waste, you get a solid waste permit. If you're 50% or more of biosolids, you go to water resources, class A residuals. And then if you're 50% or more of animal manure, then you do Vision Water Resources, animal feeding operations. And again, that 79 animal waste digesters, those are primarily on site from covered lagoons, and they're producing biogas for the renewable energy industry. And we only have one of those animal feeding digesters that is co-digesting food waste. And again, this is the same information, but just broken out into a graph. And again, I kind of paired our animal feeding operations with our one solid waste anaerobic digester, and then I lumped in our Class A biosolids facilities with our Type 4s because our Type 4 facilities with solid waste can also accept biosolids. But as you see, Type 1 facilities are the most yard waste. We do have a yard waste ban in North Carolina, and we are about to implement a lithium ion ban for batteries in 2026. And so, as you can see, Type 1 is the largest amount of facilities. Anaerobic digestion is 2. But if we just look at our Division of Waste Management solid waste section permits, again, the majority is Type 1, and we only have one anaerobic digestion facility that has a solid waste permit. And they're actually in the process of closing. So I think it could be a really great opportunity. We do have another AD facility coming online in 2026. Divert is coming to the state. But again, about only half of these facilities can accept food waste. And my office, we map out these facilities, and we recently added the exempted facilities that we're aware of. Those Type 3 exempted facilities do not have to notify our solid waste section as long as they're following the rules. If they do have a complaint, they'll likely be asked to get a permit, but we did start mapping those. We highlighted the facilities that reported food waste in their annual reports, and we also added the Division of Water Resources biosolids compost permits. And there is only one animal feeding digester on this map because it's the only one that co-digests food waste and accepts material from outside sources. So that department, they have their own map with all the digesters across the state, but we thought it was just beneficial just to report the one or show the one that just co-digests. And our office also looks at tipping fees, and on average, tipping fees are about $16 cheaper than our landfills. And our facilities reported processing over 700,000 tons of material, and as you can see, they have a variety of feedstocks. And if you look specifically at food, our solid waste facilities reported over 90,000 tons of food waste. And this has definitely increased by 200% over the last decade. So we're seeing more food waste being reported by our compost facilities, and we're really excited about that. And then we also, I've started looking at the biosolid facilities, and they reported processing over 26,000 tons of sludge mixed with about 26,000 tons of amendments. And we do have 2 biosolid facilities that are mixing in food waste into their process. And so also really want to track those. And last year they reported 2023 reported processing 490 tons of food waste. And I know this is much smaller than our solid waste ones, but I do think it's worth tracking. I have noticed a decline in those amount of permits from biosolids facilities. So definitely going to keep tracking our food waste there too. And then we're also, tracking our products for the state. We want compost to be used just as much as y'all to increase soil health, reduce runoff. And our Division of Waste Management facilities reported creating over 600,000 tons. And I know this is kind of like a busy graph, but we have, they report what's used internally, sold to the public, given to the public, stockpiled, disposed of, or other. And here's also kind of a crazy graph, but what I want y'all to get out of this is that our mulch sales kind of fluctuate, whereas our compost sales have increased over the years. And we're also looking at the biosolid products that they report. They have to, say how many Class A residuals were distributed. And On average, our facilities distribute about 36,000 tons of products a year. And we're not only looking at, the organic recycling data, but also the public programs. And we support our colleges and universities to improve and expand their recycling programs, share best management practices, and really help build out their food waste diversion programs. And it's not mandatory, but colleges and universities can complete a voluntary report to us. And they reported over 4,000 tons of organic material. And here's that same information, but just over time. And as you can see, there's a huge dip from COVID. But what I want to point out is that light green, the organic materials, has been increasing over the years. And just from this past report, we saw a 64% increase in food waste that was recovered, and that is excluding food donations. We also saw an increase in yard waste recovered, and the most popular collection method is post-consumer food waste collected in the dining hall. We're going to continue to support them. And speaking of a model program, NC State is a great example, and we love to show off this campus. Their waste reduction and recycling office helped divert 70% of the waste that's produced on campus, and they have a closed loop organics recycling facility here on campus, and they use the finished compost on their campus and the Lake Wheeler Road field labs. So feel good about using the compost bins here. Know that it goes just down the road and is composted on site, and the material is used here on campus. Also really love that they have a way to compost your paper towels in the restroom. So really, really great that we can show them off. They're a great example. And then our office, we did receive a solid waste infrastructure and recycling grant from EPA. And we're going to do, two big, initiatives with that. We're going to do a materials management optimization study. We're going to do a statewide characterization study, MRF, hub and spoke analysis, and market assessments. And then we're also going to support a statewide education campaign. But why I brought this up and why I'm really excited about it is the waste characterization study will finally give us a North Carolina specific food waste data for our landfills. Primarily, I've just looked at EPA, the national, but I'm really excited to have just one that's specific to North Carolina. And we're also going to incorporate other waste characterization studies. We are conducting 3, one in New Bern, Greensboro, and then Watauga County outside of Boone. And my office, we also provide grants. And as of 2022, we released a food waste reduction grant. We've awarded over $1.1 million in 3 years to 36 projects. And we've diverted over 40,000 tons of food waste, saved over 1 million meals, and created 7 jobs. And our RFP for 2026 is available now. So it's for recycling businesses, local governments, and then also nonprofits for like the food recovery piece. But I'm really excited that the state can provide grants for recycling businesses. I do think that's pretty unique for a state agency. A lot of them do just focus on local governments and colleges and universities. So I'm really glad that we can provide that for our recycling businesses and really grow not only the processing, but the collection and hauling. It's one thing to have a facility, but if you can't get the material to the facility, it's really a challenge. So really working to expand that. And we're also tracking our collection and exempted facilities. So again, those exempted facilities don't have to report to solid waste. So I'm doing my best just to keep tabs on where those are across the state. And a lot of our local governments have become really creative with collecting food scraps when we don't have that collection and processing infrastructure in place yet for them to do curbside. We have seen a huge increase in food scrap drop-offs across our state. We have about 51 drop-offs across North Carolina. We have about 20 food waste haulers that are dedicated to collection of food waste, and about 7 of those exempted type 3 facilities. And we're about to release this map, but we want to show where all the food scraps drop-offs are across the state. And we did break it down into 3 different categories. The red is kind of more backyard style, meat-free. And the one in the yellow is can accept meat, commercial compost, but those local governments have opted out for no compostable packaging. And we have seen more increase of local governments just saying, no, we don't want that. And then the food scrap drop-offs in green collects, meat, all foods, and compostable packaging. And then our grants did help fund the Carry Compost program. And it's a great model program here in the state. Again, if you have time, you can go by and see it. But they now have 2 drop-off centers that have collected over 5,000 pounds of food to date. And they bring that material. They use compost now, and they bring it to Brooks Composting Facility in Goldston. And then what's really great about this program is they earn finished compost through working with Brooks and Compost Now. And they've delivered over 20 tons of finished compost to Good Hope Farm. This is a historic farm in Cary, and it has produced over 100,000 tons of produce. It's about 30 acres, was established in 1910. I've been doing organic agriculture for 9 years. And Cary also recently completed a food waste collection feasibility study that was completed in 2024. So you could check out that collection feasibility study on their website. And again, my department, we not only want to, provide the grants, but we also want to have a statewide message that's similar, has the same look. So we do offer free customized outreach materials for local governments and even the businesses. So even the small local governments that don't have support to have like a public information officer or, it's really small staff, we can help provide those free graphics for their residents. And then here's just, more examples. And then finally, we did not only, again, we want to get this material composted and recovered, but we want to prevent it from happening in the first place. So we did release a food waste prevention campaign called Use the Food NC, and that just gives some tips on to residents on how they can, shop better, plan, and then also how, more information on food donation and how that is, you're protected. But thank you so much for having me. And let me know if you have any questions.
I noticed one of the graphs looked like there were some facilities that were seeing a lot more food waste, but then like total products coming onto the market was pretty flat, hadn't changed a lot. So like help me kind of square that. What do you think is going on?
Yeah, as far as like the compost products, and I think it could even just be with getting better data in our reports. So that's one thing that working with our new solid waste section, she does composting permits, and I really want to make sure that the composters that are filling out those reports are giving us quality data. So that is one goal of DEQ that we're going to work on, kind of working with the composters directly, making sure that they're filling it out, they know how to, and that information is then quality checked by us.
Sure.
Yeah. But I'm working with the data that I have, and but excited that we have some fresh energy within solid waste to make sure we have great reports.
Yeah, so I use Compost Now in Durham. It's pretty cool because they pick up the waste every week, our food waste every week from our house. It seems like they're growing, but I'm just curious from your perspective, like, are there other groups like them in the state, and do you see that model kind of like picking up from the homes working other places in the state?
Yeah, definitely. We've seen definitely increase in haulers, and we're trying to, grow the competition. Compost Now is kind of like the only game in town for now, but through our grants, we're trying to like increase more haulers and, haulers that are in harder to reach locations like the mountains, the coast. There's definitely gaps, really big gaps in our state. But yeah, I definitely see more of those entrepreneurs coming online, and I'm really thankful that our grants not only fund the like big composters and large haulers, but also the small up and coming guys too. Um, but yeah, that model is definitely increasing, and they're getting creative. I've seen some really creative ways to move buckets in personal vehicles.
Yeah.
You go more into any policies or programs to encourage use of compost? I know Duke University, for example, had it in their spec for their operations. I mean, there's the diversion side, but can you go more into the promoting the market for use?
Yeah, I think that is definitely an area that we need to grow, and I'm not sure if we have the right political climate for a lot of policy right now, but I could definitely see us trying to work with like DOT and get more local governments to use it on their site, parks and rec, football fields. So I'm not sure if how much policy we can pass here in North Carolina. But I know the North Carolina Composting Council, they have a advocacy group committee that is working on that. But yeah, I definitely think that I want to work on more resources for like compost testing and making sure that those quality products are getting out there and the materials getting used.
Yeah, excuse me, I just had a quick question because I'm thinking kind of like what you talked about in terms of preventing the food waste in the first place. Does North Carolina do any like partnerships or incentives or anything like that with sort of, I guess, the middlemen, the grocery stores, the restaurants, and those places to try to reduce food?
Yeah, our department is trying to make those connections and support them donating as much food as possible. But our businesses are not required to report to us. So I think that's where some of the information is lacking. Local governments are required by law to report information to us, but not our businesses. So I'm really hoping that some of these businesses and grocery stores can voluntarily do that with some of the coalitions that are happening at ReFed. But yeah, we're working to just, again, make those connections and, help them as much as possible, but on a voluntary basis.
Questions?
Yeah.
In terms of the map, you said it was going to be released soon. Go back to that. And how are those locations selected or come about?
Yeah, those are just the drop-offs that the local governments have reported starting, as well as the private businesses that offer food scrap drop-offs. As you can see, this is probably where most of our post-consumer food waste is collected. We had, the map beforehand with the facilities is all over, but that they don't have to take post-consumer food waste. So I think that's going to be some of my next initiative is not only to understand all the facilities, but how many are actually willing to take the food waste from the public.
Okay.
And
that's currently where private haulers fill the void, right? Is that what you're saying?
Yeah. And we do have one local government that's doing in-house collection. The city of Durham, they launched a pilot for curbside, but I think it's around like 700 homes.
Okay.
And their food waste is brought to their water resources biosolids facility. Yeah.
And in terms of, I think the other one you showed of, sort of the product distribution, like I think the biggest bar was products sold to public, right?
Mm-hmm.
And so is that sold out of the facilities, or like what is the model there? Like, is it one centralized point where public come to?
We don't track that in the reports, but my guess is a variety, like directly from them, but also at garden centers,
Okay.
yeah, in bulk too. So, but we don't currently track that. Yeah.
Anything else for Christine?
Quick question. So I work at a utility in Greenville, South Carolina. And I came in a little bit late, so you might have touched on this, but where do you see the pros and cons and like what fits better in terms of like anaerobic digestion and diverting organics that way compared to composting? Like where I'm sure for the source might make sense for one or the other, how the nutrients are used on the back end. So Just curious where you're seeing that going.
Yeah, we don't have much food waste anaerobic digestion
Okay.
here in the state. We only have one that's currently coming offline and another one that's coming online.
Okay.
And then we have, I guess there's two, so really just one right now. But I do want to see that grow because I believe that we need all the help we can get. I'm not pro compost or pro AD. I want them both because we need to, as much as possible, to address this problem.
Okay.
But I do think that I tried approaching our like hog farm lagoons that are doing AD, and I've come to realize like they don't want to co-digest. Like they like that consistency of that manure, and so I think again AD is something I'm trying to learn more about too. Came in this role more as a composter, so yeah. But we definitely need all the help we can get. Yeah, and if there is anybody that knows of like a company that would want to, like there is some current infrastructure, that one anaerobic digestion facility that's coming offline. I'm not sure where it is, but there is, a lot of infrastructure in place that I think another company could come in and, take that over. But it's in Charlotte. So if you have any questions about that, would love to try and get that used properly and keep that infrastructure in place. So you can talk to me afterwards.
I have a question. Why that facility is coming offline?
I do not know. Yeah, I don't know why they're stopping operations.
That was based.
Yes, yeah. And that really disrupted a lot of our haulers that were taking material there. So I'm glad that, we do have some small haulers in the Charlotte area, but nothing compared to what that facility was processing.
And so our next speaker is Doug Call, Associate Professor of Environmental Engineering here at NC State. His group focuses on resource recovery, nutrients, and energy using biological and electrochemical technologies. He's active here in the STEP Center. His group working with water resource recovery facilities, understanding biological phosphorus removal in particular, how it works well, control strategies, and really to prevent the sort of unstable behavior that occurs. And then today, Doug is going to be talking about some work in collaboration with Bruce Rittman. I'll let him elaborate on that and looking at the phosphorus speciation during digestion. So thanks, Doug.
All right. Thanks, Trevor. Great to be here. As Trevor mentioned, I'm actually filling in today for Bruce Rittman. He couldn't make it. So a lot of the work you're going to see today comes from his group. So I'll do my best to try to describe it. Hopefully he's not watching online and you're going to send me an email after this. Yeah, make sure kick him out of the room. Myself here at NC State, along with some of his students, Maheen, contributed a lot to this work. One of my students, Jenny Ding, I'm going to mention her work today, studying phosphorus speciation. Joshua Boltz, who works with Bruce as well, who is currently working at Woodard and Curran, who has done some really nice modeling work that I'll just touch on and unfortunately won't be able to go into detail. But as Trevor mentioned, we're a part of the STEP Center. And one of the big things that we've been wondering about since the formation of STEPS is what shapes and forms and different species of phosphorus exist in different matrices, in particular organic matrices. And so I'm going to spend most of my time touching on that, and then also talk a little bit about some work that Bruce is doing to try to recover phosphorus from these high-strength organic streams. So I'm not going to say too much here. I think we're all on the same page in terms of this mass balance. If you're a member of STEPS, you have dreams of this because we have seen this all the time the past couple of years because it's so important, right? It's figuring out where the phosphorus is going. And the key takeaway that I want to highlight here in red is that a lot of the lost phosphorus, we could call it, is associated with organic wastes, whether it's animal manures, food processing, which we've heard about from previous speakers, and human excrement as well. And so if you add those up, roughly, we're talking about 50% or so of the phosphorus that comes into this mass balance ends up leaving as these wastes. And so here's some images of how we might receive these, right? So we got a nice big bin of food waste. We've got a semi-happy cow standing next to some cow manure. It's these kinds of large sources of organic waste streams that we're particularly focused on in this project. Another thing we often talk about in STEPS is, should we just focus on getting phosphorus, or should we think about maximizing the potential recovery of all kinds of things that might be of value in different wastes, whether it's nitrogen as well, whether there's valuable metals, what technologies exist, or maybe that we could invent that could help us recover more than just the phosphorus piece. And so that's where Bruce's group, my group as well, we focused on a technology we've heard about already, anaerobic digestion. Why? Because we can get nutrients out of it, phosphorus, nitrogen, but we also get biogas, right? And so that can be a resource that can help potentially bring in some additional economic support for the technology. We've heard a lot about some of the other benefits as well, so I'm not going to touch too much on here. Things like reducing biosolids, so less going to landfill, improving soil health, and so on. So there's multiple benefits. And so that's why we have focused on this technology. So I'm going to break this down into really two key questions that this project has been focusing on. One, what forms of phosphorus are generated under the anaerobic treatment of organic waste, so such as those digesters in the previous photo? And then the second piece is, okay, well, maybe we can release the phosphorus from some of this organic material. How do you now recover the phosphorus? The second part of the story. So let's start with the first one. The main take home here is, when we think about organic waste, phosphorus can be associated with that waste in all kinds of different forms. I think many times when we have discussions about recovering phosphorus, we often first think orthophosphate, right? So the PO43 minus. And so we think, okay, how do we, for example, design a sorbent that can attract that species, right? Or just phosphate. Sometimes that's the term we use to describe this. But when you look at those pictures of these organic wastes, and you dive really deep down to the molecular scale, this is likely what you're going to see. All kinds of different organic forms. These are model compounds. So top left, on this right-hand side, that's
Pointer
oh, thanks. So the top, perfect. Right up there, that's phytic acid. That's associated with a lot of plant materials and seeds and so on. So food waste is going in, you're likely going to have that material. Glucose 6-phosphate. So microbes, when they will bring glucose into their cells, they'll attach the phosphate on there so that it sticks around and doesn't just escape out of the cell. So you'll have that material. This is a representative compound of a lipid, right? So phospholipids, for example, they have phosphates in there. And then everyone here, I just probably knows this molecule. I just taught this in my class the other day. This is ATP, right? A big energy molecule in pretty much all living material. Okay, so you can see the P's in there, right? They're all scattered about. And so that looks nothing like this here. Why does this matter? Why do we need to answer this question? Well, really, there's two reasons. It influences what strategy we're going to use to try to recover that form of phosphorus, right? Like I said, if you're designing a sorbent for phosphate, and then you apply it to try to recover phosphorus from ATP, it might not work. And then the second piece is thinking about, if we do recover these forms, how bioavailable are they, for example, if we're using those as fertilizers? So we need to really understand what the speciation is. There's pretty well established protocols and guidelines, stepwise procedures to try to figure this out. They're not perfect, but in general, what we do is you say, let's take the total phosphorus in a sample. Let's fractionate it between what's soluble, so what's dissolved. And typically what we do is just pass the sample through a filter with little pores in it. What goes through is, we say, dissolved. This could be organic, like I mentioned, some of those molecules on the previous slide, or it could be that orthophosphate. the non-dissolved fraction is the particulate form. So these would be the solid things. Maybe you might even see them floating around. Similar, those could be on the organic side of the story, or they could be on the inorganic side of the story. Inorganic, maybe it's phosphate bound to things like metals. And then within the organic, this is where things get really complicated. And I think you'll see later where most of the question marks exist. It could be material that's intracellular, so inside microbes. Maybe it's extracellular, so microbes and digesters, when they're growing, they like to spit all kinds of big molecules outside of their cells. Many of those have phosphorus in them. So it gets very complicated. So what do we do to try to answer these questions? Well, really, there's two major projects. I'm going to spend most of the time talking about what Bruce's lab has done at Arizona State University, where he's worked on doing bench scale studies of dairy waste fed systems. At first, when I was looking through his slides, I was like, oh, it's feeding it milk, waste. No, it's manure. It's manure from cows. And looking at different variables, like hydraulic retention times in the system, to understand the P speciation. And then the second thing I'm going to talk about is work that my group has done, where instead of putting in a very complex waste, we said, can we put something in that represents an organic compound and more carefully track it, right? So let's put in that phytic acid, and let's compare that to if we put in a lipid, a phospholipid. So I'm going to talk about those two experiments. All right. So just to jump right into, really, I think this is his key findings. And what you're looking at here, let me step you through this. You've got the relative percentage of the phosphorus in both the influent and then the effluent. And so that's the control, and that is just the anaerobic sludge. So the anaerobic sludge, that provides the microbes. When he combined them, so here's the anaerobic sludge plus the dairy waste in different ratios, and then looked at different retention times. So some variables there. Each time you've got your influent, and then afterwards, at the end of the experiment, you've got your effluent lines there. These are broken down. So how we do this is, again, there are somewhat established procedures. They're not always great, but they can help you differentiate. For example, you can figure out what are the orthophosphates. So if you just take a sample and do the standard test for orthophosphates, the reactive phosphates, we consider that those are the free, not bound orthophosphates. Then you can do additional tests like some acid extraction, some alkaline extraction, some temperature changes and things like that to your sample to try to tease out what these might be bound to, or if we've got an organic fraction in there. And so that's what you're seeing here. So for example, here's the orthophosphates, the easily exchangeable phosphates. So some of these materials, they might be bound, but they're not bound to something like a metal that is really tight. They might be loosely bound with organic material, and so we can freely extract them. And then the other ones in the green, and then the gray lines here, these are attached to different metals. So it's iron and aluminum, and then calcium and magnesium. Then there's the final piece, the dotted box there on all of these samples. These are other, right? And we don't know what they are, but based on doing this fractionation, it's presumably organic phosphorus. What species? This is what we don't know. All we can say is we're pretty sure it's an organic form. So what's the key takeaway from this? One is that most of this phosphorus that is released, so that occurs at the end of operating these digesters, even across different retention times, we pretty much get it locked up. It's still locked up in inorganic forms, and then this unknown organic fraction. What we're trying to do, and really what Bruce is doing here now, is trying to model this, right? So model the breakdown of this material, because if we can model it and it agrees with what we're seeing experimentally, then it shows that we really understand what's going on, both in the release of the phosphorus, but then the fractionation of it. There's a lot of information to unpack here. I'm not going to go through this, but the main thing I want to point out is he's working on both an electron balance and a phosphorus balance. So as electrons in the food are being degraded and go through different processes, so the formation of carbohydrates, formation of proteins, and those extracellular polymeric substances, how are those electrons being distributed through those different processes? Ultimately, ending up with some of the different fates of those, whether it's volatile fatty acids, biofilm associated compounds, and then reactive ortho P as well. So really what the novelty here is not just completing the electron balance, but putting in the phosphorus piece, which has really been missing. And so he's taking some of the experimental data that his group and my group have been collecting to try to fill in the red spots here and close that gap in terms of phosphorus. One kind of interesting thing, this is really preliminary, is based on this model, what they predicted is that roughly 20% of the total phosphorus would be organic. And what's, like I said, interesting is if you look at the experimental results in the effluent, it ranged a little bit, but we're somewhere around 20% was this unknown organic fraction. What's kind of neat about this model is it can start to help us understand what those species might be. And that's what you see here, is all of this is at 20%. And based on their model predictions, this is where they think some of this phosphorus may have ended up. So for example, some in the protein-based extracellular polysaccharides, endogenous decay products. So as cells will start to eat themselves, they'll release materials. Carbohydrates in the EPS. So we can start to better understand what some of these organic compounds are and where that phosphorus might be. So some of the work that we've done, these are just some highlights of the tests that we did. Ours was different. We asked a similar question, but we went at it a different way. Instead of putting in a very complex waste like manure or food waste, we said, well, let's just put in a very specific compound, organic or inorganic. And I tried to squeeze them in here. I know they're hard to see, but what we tested was aluminum phosphate. So add that as your phosphorus source. Glucose 6-phosphate, lecithin. So this is a representative phospholipid. And then the phytic acid, which is over there in the corner. So we fixed the phosphorus content. This was one we had to figure out. Do we fix the phosphorus content, fix the COD content? Because a lot of these molecules, they have their own chemical oxygen demand, their own COD. So it makes it a little bit challenging. But what we decided to do is they say, let's just fix the total phosphorus content and say it's coming in different forms. And so that's what you're seeing here in an anaerobic digester over a period of about 22 days. Well, the first result wasn't too surprising. The aluminum phosphate just stays bound up. Right? You put that in the digester over 22 days, basically none of that phosphorus is released at all. And that's to be expected, right? It's really bound tightly. Maybe there's some microbes that might have enzymes that can solubilize a little bit of that, but it's not going to release a huge amount of that phosphate. Why did we do the aluminum phosphate? Well, many facilities in the wastewater industry, they'll use alum. They'll add aluminum in order to precipitate out the phosphate. So this represents, a potential source of phosphorus that could be going into a digester. Let's go to the organic compounds. So first of all, the glucose 6-phosphate, and what you're looking at with the different colors are different fractions. We've got the definitions down here. So we either have loosely insoluble organic phosphorus, loosely bound orthophosphate, loose organic phosphorus, and then solid total phosphorus. So different fractionations of both the organic and the inorganic. And what you notice is that the glucose 6-phosphate, pretty much right on day one, just gets all released. I think one of the reasons for this is it's a small compound. The microbes are already eating glucose and they see this, and so they've already got the enzymes ready to go. They're not, in my opinion, they're not after the phosphorus, they're after the carbon. And so really they just want to chop this up and get that phosphorus, in a sense, out of the way so that they can get on with using the glucose. But you notice that it goes really fast as a release to that loose ortho P. why you're not seeing a lot of this end up in a solid form, like an inorganic solid, is this was a very cleaned sludge. So we got this from an anaerobic digester, and we cultured it in the lab to clean out any residual phosphorus. And so we don't have a lot of metals in there to begin with, like you see in the manures and things like that. The other compounds are a little bit more interesting. So the lecithin, this is a very big compound. So if you think about food waste that might have fats, oils, and greases, these things are big. So the microbes are going to have to excrete enzymes outside their cell to start to chop this thing up. And so we see a delay in terms of the breakdown and the release eventually of the orthophosphate form. Again, this is an excess of phosphorus for them. So that's why we see a lot of it accumulate. Really, what's more interesting in this study is just the fact that we see differences in the delays of how these compounds are broken down. And then finally, the phytate or the phytic acid there, it's a similar story. It takes a little bit of time for microbes to synthesize the enzymes to break these down. What is kind of interesting was that we did some enzyme tests to look for phytase activity, and we did find a good bit of activity in the anaerobic digester, which, with talking with people that work in the soil field, said that's pretty interesting that you've got that much activity in an anaerobic digester, because in many cases it can be difficult or a little bit challenging to find those enzymes to break these materials down. Was it bacteria? Well, we did some basic sequencing to try to fingerprint who's there. The bacteria we did find didn't have the phytase enzymes, at least based on associating what we know about them in the literature. Of course, we could have different strains in our bioreactors, but we did find a lot of fungi, a lot of fungal communities that we know nothing about. And so this is something we're looking a little bit more into, the potential for the role of fungi inside of anaerobic digesters to contribute to some of these phosphorus transformations. Okay, so that's a little bit about the forms. Let me touch just a little bit on the recovery, because again, this is more out of Bruce's group. He has teamed up with some researchers at Northwestern that had developed this Pearl system, right? So now let's imagine we've released the phosphorus from these compounds. How do you recover it? And that's where they envision that this Pearl system comes into play. Phosphate elimination and recovery lightweight media. It's a good one. And essentially what they do is they start with this cellulose sponge. And through methods that they use in their lab, they're able to coat magnetite, roughly 6 to 16% on a mass basis. They dip coat this. So you've got essentially a nanoparticle covering all over the sponge. You run this through phosphate adsorption experiments. They're still figuring out what the exact mechanism might be. So you might have just simply electrostatic attraction. So just based on the charge differences, that might pull that phosphate close to the iron, or ligand exchange. So you got the hydroxide group that pops off and a phosphate pops in. You get the adsorption, and then here's one of the big challenges with really any adsorptive material when it comes to phosphorus, is how do you get that phosphate off? And so what they found is that by taking this material that has the phosphate absorbed, they can put it in a base solution, it releases the phosphate, and then they can redo another cycle. So that's the vision, is this adsorption, desorption cycling. They've done a bunch of tests with simple, clean solutions, but I just want to jump to this one, where they have tested real anaerobic digestate. What you're looking at is the adsorbed P, so milligrams of P per gram of this material, this sponge. The P concentration, so your P concentration is the orange arrows, and then the amount of adsorbed P. And so you see this word cycles. What this means is they adsorb, then they do a desorption, and then they take the same material back and go for another adsorption cycle. The main conclusions so far, and they're still, again, trying to figure out the exact mechanisms, how some of these variables like pH affect this. But generally what they've seen is that, one, lowering the pH tends to help. And they think what that is helping to do is give this material a little bit more positive surface so that it can help attract the phosphate. When they've done some of these absorption cycles, they've gotten upwards of 90% removal in terms of the phosphorus. The desorption piece is a little bit more challenging, and I think that's what they're focusing on now. pH plays a huge role. So, for example, increasing from pH 9 to 10, you can get a desorption efficiency from 54 to 63. So they're looking at both pH issues, but then also modifying this material so that they can try to desorb at lower pH. One of the main goals here being you don't necessarily want to have phosphorus in a really alkaline, salty solution. Can we lower that pH so those demands are lower? All right, so let me wrap this up. Really the main conclusions here. What are the major forms of the phosphorus generated during anaerobic treatment? Really, the answer is a mix, right? We certainly can't say it's just orthophosphate. But really what we found with this specific waste that was tested was mostly phosphorus is bound to metals, and also it's bound up inside of organic materials. And then how do we recover? Well, like I mentioned, these iron-coated sponges initially in these preliminary tests show promise for this ability to do multiple adsorption, desorption cycles. So I'll finish there, and just recognizing that this work was funded by STEPS.
We have time for a few questions.
I'll try my best.
I was wondering if you could go back to the slide that had the different species before and after.
Yeah.
Right there. So this is sort of from a nutrient management plan perspective. It looks like in all cases, and tell me if I'm wrong, that essentially if the material was applied to land before versus after digestion, you would have less plant available or less easily available, water soluble, loosely bound up phosphorus after digestion than before.
Just based on the differences between those two influent effluent,
Yeah.
yes, but your product is going to be much different too, right? What goes in versus what comes out, the solid material, you're going to destroy a lot more. There's a lot of things missing here, right?
Yeah.
Destroy a lot more of the volatile solids. You may even dewater a little bit inherently. So
Yeah.
it's not a full apples to apples.
The concentration of phosphorus, of total phosphorus in the effluent is going to be higher than in the digestate than in the influent.
Yeah,
because
right.
you've burned off organic matter. But what I'm wondering is, most of the states that we work in when we're doing nutrient management plan with digestates, they're just looking at total phosphorus. That's essentially how we're regulated.
Right.
And I always wonder if a case could be made that we're actually adding a little bit less, that the percentage of that phosphorus that we're adding is less plant available, more likely to be bound up in inorganic form or some other form in the soil than if that material had been applied before digestion. So I'm wondering first if that's true, and secondly, once it's added to the soil, does that all change again? So it might be more apt to be desorbed after being added to the soil.
Yeah, those are great questions. I don't know the answers. My guess is that there is a good question about if you were to land apply this now, what's the bioavailability? What's the time scales of phosphorus release from these different solid compounds?
Yeah.
Don't know. I don't know. I mean, the soil scientists might know. I mean, I think in general, my understanding is a lot of these phosphates bound to metals.
Yeah.
They can be pretty locked up in there.
Yeah.
Right. The organics, maybe there's a potential there for faster release. If you have, active microbes in the soil, they can break those materials down. Those are just guesses.
Okay.
Yeah.
Thanks.
Yeah. Thanks.
Our next speaker we have is Amir Varsashovi. Since organizing the founding, he has leveraged his technical expertise and unrivaled commitment to guide green technologies to the pinnacle of success during his tenure as Chief Executive Officer. Prior to founding Green Technologies, he devoted his talents and time to academia while earning his PhD in soil and water science from the University of Florida. He's worked as a research scientist evaluating commercial fertilizers for efficacy and environmental impact. So welcome, Amir. So I want to give Matt some credit for Matt Schultz some credit for this sort of final set of slides. He thought it'd be great to connect this session to what comes very soon, which is lunch, right? And so, I have just a few slides pulling from some literature and a little bit of work that we've done, thinking about the phosphorus footprint of our lunch, specifically, and, our diet kind of more broadly. And so we start with this paper that Jim was part of a little while ago, thinking about the role of diet in phosphorus demand. And, in this work, they really worked through some calculations that we could get to something like a phosphorus footprint, right? And so looking at, if we think of the food that we're going to have at lunch, right, it's going to be, some animal products and, just, vegetable agricultural products. And so looking at, animal operations, the feed that goes into them, what is the feed composition? What is the phosphorus that's within that feed? And then what comes out of that would be various conversion factors that think about, how the phosphorus is applied to the land, what's grown, and then how that moves through the food chain. And so we're going to start with sort of that, just with a couple slides, and then go from not only thinking of phosphorus footprint from a quantity perspective, but then sort of end thinking about phosphorus footprint from a quality perspective as well. So I'll elaborate on that. So here I'm just highlighting a few things in terms of, the food groups that were here. And, in terms of the context of our lunch today, Matt told me the menu. And so we're gonna have some vegetables in our lunch, some beef, some chicken. And so we can kind of follow this through and we get to this factor here of this conversion factor of kilograms of phosphorus per kilogram of crop or animal. And we can start to see just from this factor, right, that beef, of course, has a big phosphorus footprint because of the feed that goes into it. And obviously, the vegetable would have the lowest phosphorus footprint, the chicken in between. So that's sort of the quantity piece of the phosphorus footprint. Jim is a co-author on this work, so he can kind of walk you through the finer details. But I want to take that phosphorus footprint and now pair it with, even more recent work, which is, really getting into precision nutrition and diets. And just as a reminder for us, Doug showed some of the kind of macromolecules that phosphorus is part of. A big part of our diet is proteins, right? And so proteins are, if you remember your biochemistry, strings of amino acids. And, what happens is you bring in proteins into your body breaks down those amino acids because in our human metabolism, we need amino acids for various functions within our body. Some of those amino acids we can create. However, there's essential amino acids that you can only get from your diet. And the idea of this, precision nutrition would be really starting to think about our diet down to the sort of essential amino acid level. And so what this paper and other papers have done is develop a variety of frameworks that, look at your diet and start to break it down all the way to the essential amino acid level and then create like a score. And so just kind of showing a highlight here, this is from one result from the paper. It shows up a little small, but what we have here along the bottom are essential amino acids. And then, we have something this was a sort of a meal that they put together of rice, milk, cinnamon, blueberries, walnuts. I guess they're putting together maybe a breakfast or a lunch. And then we can see that there's both a variability in terms of the essential amino acids that are present and the amount of amino acids that are there. Right. And so what we could start to do then is, couple what we saw from the first work in terms of actual quantity of phosphorus footprint, and then overlay on top of that a quality component in terms of the essential amino acids that are there. And so if we bring all that together, and again, Matt helped with these calculations, Jim was part of these calculations, we can now look at our lunch menu of our herb-baked chicken, our smoked beef brisket, beans, rice, vegetables. And we took the kilogram of phosphorus applied, and so we can see that. The next column comes from the USDA in terms of protein digestibility, corrected amino acid score. And then we get this final column, which sort of tries to bring together both the quantity and quality piece, which is this kilogram of a digestible protein per kilogram of phosphorus applied. And so if we look at that, what we can see is, a higher value there is good, right? You're getting, digestible proteins that are good from a nutrition point of view. You're sort of maximizing that relative to the phosphorus input. So, we see the chicken and we see the vegetables coming out kind of as winners, and the beef maybe being, it was less efficient to start with from a phosphorus footprint, and it's not kind of giving us those essential amino acids as well. And this is just, I'm not a nutritionist, I'm an environmental engineer, for a full disclaimer here, but it's starting to give us some insights. But, as an environmental engineer, I have kind of one more slide to wrap it up. Doug and myself and others in environmental engineering and within STEPS, have a major focus on phosphorus recovery. And that came out of today's talks as well. And so what I've wanted to try and do is really connect that phosphorus recovery all the way through sort of the food supply system. And so what we've done is a life cycle assessment. I don't have the schematic shown here, but essentially with a life cycle assessment, we looked at two scenarios broadly. We looked at producing a beef burger, and we looked at producing a soy burger. And so for those two systems, we looked at all of the energy and material inputs to those two systems. And what we varied was just the phosphorus input. We looked at having commercial phosphate fertilizer go into that food system of producing a beef burger or a soy burger, or look at recovered phosphorus. And so you can, and so what you get out of an LCA are these midpoint environmental impacts. And the idea is that these midpoint environmental impacts connect to eventual sort of outcomes in terms of, say, human health or environmental health. And then you look at a relative impact of these. And so the higher the bar, it means the higher impact it's having in one of these categories. What we could have a discussion about is, which of these categories are more important, for example. And so these are different midpoint categories. This is the relative impact. The red relates to beef burgers. The blue relates to soy burgers. And then when it's, the diagonal lines, that's the recovered phosphorus. And for us, the recovery process is from human urine. So you can find a lot of literature that will tell you, of course, a soy burger in general has lower environmental impacts than a beef burger. But what's really interesting is if you look at the red sort of diagonal bars, they're always lower than the solid red bars, and sometimes quite a bit lower. And this comes about because we have taken recovered phosphorus and brought it into the food chain instead of using conventional, mined phosphorus. You don't see as dramatic of impacts in the soy burger. You do see those diagonal blue bars being lower. But, the phosphorus footprint, as we saw, going into the beef production versus the soy production is quite different. And so I think this is promising, I guess, if you're going to have the beef brisket for lunch, you could pretend that it came from recovered phosphorus product, right? And what I could say now is that we're repeating this work, bringing in the nitrogen recovery, because it came up about just about everybody today that, nitrogen is a big piece of a lot of these waste streams. And so when you bring in the nitrogen recovery, we've just done some preliminary work on that, you see an even bigger reduction. And that reduction then is also carried through in both sort of meat-based and more, sort of vegetarian options as well, because of the role of nitrogen in both of those. So I thought this was a great session, and hopefully this kind of brings it all together in terms of what the recovery can actually do within that food supply system. And we are right on time for lunch. All right, so thanks, everybody.